The overall goal of this protocol is to generate tolerogenic and mature dendritic cells from monocytes for experimentation or vaccine development.
This method can help answer key questions in the field of immunology by providing a model to study the development, maturation, and antigen presentation of dendritic cells. The main advantage of this technique is that it enables the generation of high numbers of dendritic cells in vitro for experimentation.
Begin monocyte enrichment by mixing 20 microliters of previously prepared peripheral blood mononuclear cell, or PBMC cell suspension, with 20 microliters of trypan blue, to count the number of live cells using a cytometer. Then, centrifuge the cell suspension. Aspirate the supernatant completely, and resuspend the cell pellet to a concentration of 80 microliters of staining buffer per 10 to the 7th cells. Add 20 microliters of CD14 microbeads per 10 to the 7th cells, mix well, and incubate the cells for 15 minutes at 4 degrees Celsius. Wash the cells with 1 milliliter of staining buffer per 10 to the 7th cells, and centrifuge the cells. Aspirate the supernatant completely and resuspend the cell pellet. Obtain a medium column for a maximum of 2 × 10 to the 8th total cells, and place the column in the magnetic field of a separator. Next, rinse the column with 500 microliters of staining buffer. Pipette the cell suspension onto the column and collect the unlabeled cells that pass through the column in a 15